Welcome back in another video lecture from microbiology with Chagnik. So in this video tutorial, or I should say from this video tutorial, I am going to start a new video lecture series that is on the basis of restriction mapping. So this video is just a introductory video for restriction mapping. After that video, I am going to solve various kinds of restriction mapping problems, and I am going to explain you the solution of that problem so that you can answer any kind of question from the restriction mapping topic, that whether it is asked in your university exam or any kind of competitive exam. Okay, so before going to solve the questions, just try to learn what is restriction mapping. So restriction mapping is nothing but, suppose you have a long DNA molecule, okay, very large DNA molecule. Now you need to know the appropriate recognition site, okay, that appropriate recognition site of the various kind of restriction endonucleases. Okay, so in case of unknown DNA it is not possible, but whenever you are dealing with a very known and common sample, restriction mapping of that particular DNA or genome is possible. So what is restriction mapping? Restriction mapping is nothing but a cut or the recognition site. Okay, restriction mapping is what? Restriction mapping is just a simple map of various recognition or restriction site of various restriction endonucleases of a particular DNA molecule, so that you can easily identify which kind of restriction endonuclease to choose whenever you are wanting to digest that particular DNA molecule. Okay, so that is what restriction mapping, and why restriction mapping is important. Okay, because whenever you are dealing with any kind of known molecule, and suppose from that you are wanting to exclude a gene of interest that you want to clone, okay, and now if there is no kind of information about the various recognition site that is present inside the DNA molecule, so you don't figure out what kind of restriction endonuclease to choose. That is why restriction mapping is performed to determine the particular restriction or recognition sites that are present in that particular DNA molecule, so that scientists find it helpful to figure out which kind of DNA molecule to choose, which kind of restriction endonuclease to choose, and which not. Okay, so in restriction mapping related problem, they will give you two enzyme in graduation level. I am making this video on the basis of the graduation level. We are not going to discuss about the triple digestion. Okay, so this is where I am confining my video up to the double digestion. So they will give you two enzyme restriction endonuclease, okay, RE1, RE2, and they will give you the restriction fragment generated after digestion of that particular enzyme. Okay, suppose two fragment, and they will also give you the result for double digestion. What is double digestion? That means in that time the particular DNA molecule is treated with both. Okay, and then you have to find out where a particular restriction endonuclease is present, and that particular restriction endonuclease is of whom. Either it is for RE1 or either it is for RE2. That is your job to find out in restriction mapping problems. And remember one important thing: if in any question you do not find out any kind of double digestion, then make sure that either the question is wrong or they want to ask anything else. Okay, they do not they ask you to construct a restriction mapping, and otherwise your question is wrong, because without double digestion the restriction mapping cannot be done. Okay, so that is another important point. And remember, in some cases they will give us about the vector, that means circular plasmid. Remember, if one restriction endonuclease is present, then it will generate only one band. Okay, you will have only the one band, so they do not get bothered whenever you found that in a particular plasmid there is a one enzyme working and you have one band. It is common because after restriction digestion by one endonuclease, it plasmid become just linearized, but they do not become two leads to the formation of two fragments, right? So that is the idea. But whenever you are working with a linear DNA molecule, if one restriction endonuclease is present, it will lead to the formation of two band. Okay, so from the next video I am going to discuss with you the mathematics problem. Okay.